Okay, cool. Thank you for being here. Sorry online for the delay. So this is a CGGG seminar with an invited speaker from Liverpool University, Tarang Mehta. So Tarang is coming with the CGGG here through a connectivity grant that we won a couple of months ago to invite more speakers. Tarang is a lecturer in bioinformatics at the University of Liverpool, just started there in April. His research broadly explores the genetic underpinning of vertebrate adaptive traits and disease response with an outlook towards preserving organismal health and biodiversity. And so much of his work uses multi-omics data to characterize genetic diversity and identify marker of economically important traits, so which is very relevant for the CGGG focus on our seminars. So Tarang completed his PhD at the National University of Singapore, studying vertebrate genome evolution, and then he did a postdoctoral work at the Erlang Institute, focusing on characterizing genes, regulatory network associated with adaptive traits. Yeah. Thank you, Tarang, for being here.
Thanks.
And yeah, the floor is yours.
I need to remove that chat, which I can't even see it on here. Do you want me to go over to that?
Oh, yeah.
There we go.
Yeah, just.
There we go. Cool, I think we're there. Yeah, thanks for having me here, actually. I'm really excited to be here. Funnily enough, I think kind of historically, a lot of my work has overlapped with some of the things that you have going on here in terms of kind of aquaculture research. However, this is the first time I visited the Roslyn, absolutely lovely campus. And so today, what I'm going to do is I'm going to speak a little bit around some of the work that I've been doing the past few years, kind of developing multi-omic computational approaches to studying aquaculture relevant traits. And so much of my work kind of centers around how gene regulatory network evolution can drive phenotypic change. Shall I just get rid of that as well? Which is there we go. Right, I think we're about there now. So seminal studies kind of within the 70s showed, when they were comparing kind of human and chimp protein sequences, that despite these kind of grand levels of morphological diversity you see between comparing human and chimp, that the protein sequences were actually quite highly conserved. And so at these very kind of early stages, decades ago, they were kind of concluding that the evolution of regulatory processes were really important for functional gain. Step forward a little bit further in time, and then several studies then expanded on this, providing evidence on the role of cis regulatory elements towards morphological diversity, kind of all concluding that the molecular tinkering of regulatory systems ultimately was contributing to these levels of morphological diversity. And so taking all of these like studies that happened decades ago, or slightly a little bit more recently, my working hypothesis with a lot of the work that I do revolves around rapid gene regulatory network evolution. The genotype has facilitated adaptation, the phenotype to a variety of ecological niches, the ecotype. And so to study this, more recently, I've been using the East African cichlid fish system. As you can kind of see here, they're one of the most diverse vertebrate radiations on Earth, showing a great diversity of shapes, sizes, colorations, behavior, and lots of ecological specializations. Now, in an effort to identify the genetic changes responsible for this remarkable radiation, the genomes and transcriptomes of these 5 representative cichlid species were sequenced, assembled, and analysed in 2014. And they found a number of kind of genetic mechanisms contributing to the adaptive radiations, of which then other studies took that a little bit further, focusing on more kind of the individual species within the 3 Great Lakes of East Africa. So in Lake Malawi species, they were able to find really low levels of genetic diversity between species pairs, so only 0.1 to 0.25% genetic diversity. Within Lake Tanganyika, they were finding that species richness was not linked to accelerated coding sequences, but instead that the non-coding transcriptome was evolving much faster than the coding portion. And then within Lake Victoria species, finding that ancient indel polymorphisms, again in non-coding regions, contributed towards species divergence. And so taken together, a lot of these studies were finding that the East African cichlid genomes were quite similar just in general, but ultimately implying that discrete differences, like, for example, regulatory changes, play an important role in the phenotypic divergence differences between species. And so we used a lot of these hypotheses then for studying the genetic basis of adaptation within these same 5 species that were studied in 2014, of which the genomes were generated. This time, though, using the integrative omics approach to identify regulatory networks that could be driving some of these adaptive traits. And so developed this kind of computational approach, again, integrative, to interrogate gene regulatory network evolution. I won't go into much detail in this. I'm happy to take questions on it, but essentially what we're using is we're taking kind of gene expression, species, phylogenies, as well as gene trees, integrated in a co-expression based approach, where we then actually develop a separate motif prediction pipeline. If anybody's actually predicted, say, transcription factor binding sites within non-model vertebrates, what you'll essentially find is a lot of the position weight matrices for these motifs are directed from human and mouse studies. So when you port them over to things like fish, which are evolutionary distant, you miss a lot of information, which is potentially there because we're trying to leverage really short sequences derived from humans and mouse to fish. So we essentially developed our own database of clade and then potentially species-specific position weight matrices that we can then use to scan the genome to identify specific transcription factor target gene edges that we can then build in a gene regulatory network reconstruction approach. So again, we applied this on genomic data as well as transcriptome data for 5 cichlid species. And essentially what we were able to find is instances where we have a presence and absence of a transcription factor target gene relationship. So in this instance, MR2C2 is directly regulating a visual opsin gene, SWS1, directing expression pattern of that gene in Embrycardi. However, due to a discrete variation in the transcription factor binding site, we find that this is broken within M. zebra, and we were able to functionally validate this. And we were then postulating that this presence and absence of transcription factor target gene relationships were associated with, say, water depth and dietary differences between the 2 species, basically enabling this visual system opsin gene to be switched on and off depending on the environment that these species were found. So we published that work in 2021 and then took it a little bit further, where basically we had small RNA data as well from the same 5 species, and thought, well, instead of focusing on these large, horrible network hairballs that you can generate for transcription factor target gene relationships, instead developing this 3-node motif approach to identify more meaningful interactions that, again, could be driving some of these adaptive traits that we've seen. And so when I say 3-node motifs, basically for a transcription factor, we can see that it can either activate or repress a target gene, whereas then a microRNA can then post-transcriptionally repress either the target gene or the transcription factor in this feed-forward loop mechanism. So we take basically the transcription factor target gene relationships from our previous study, and then in this study, then de novo detect, using in silico approaches, microRNA binding sites within 3' UTRs of the same target genes. And so kind of across the 5 species, we were able to identify around 80 million 3-node motif edges, and then taking 1-to-1 orthologous relationships, we were able to compare those across the 5 species. Kind of similarly, again, we were able to find meaningful interactions that could be driving some of these adaptive traits, differences that we see between the 5 species, again, focusing on the SWS visual opsin gene, we were able to identify a transcription factor and a microRNA co-regulator, which was either present or absent, one in the kind of 5 species, but then looking along a larger then phylogeny of Lake Malawi species, where blue showing that it's present and then yellow showing that it's absent. And again, this is due to discrete variation within binding sites, dictating whether we have these regulatory relationships. Essentially, what we were able to find, though, is that this discrete variation was segregating according to phylogeny and ecology of species. So those, let's say, within the same clade, sharing a similar environment, were having presence or absence, as an example here, of the regulatory relationship, and so forth. And so we study these papers, and so I suppose to just quickly summarize, in our first paper, we were able to show discrete transcription factor binding site mutations disrupt regulatory edges and are associated with gene regulatory network rewiring of cichlid adaptive traits. And then in a kind of follow-up paper showed that positive selection acting upon regulatory mutations, so these are in both transcription factor and microRNA binding sites, are associated with gene regulatory network rewiring. Again, that can be associated with adaptive traits such as the visual system. And so in these 2 studies, we focused on 5 species, and essentially we're almost neglecting Nile tilapia within these cases, because we were using it as an outgroup to then compare within adaptively radiating cichlid species. And so thought to focus then a little bit more on Nile tilapia. And whilst it's not an adaptive radiation, tilapia just in general, there are, however, more than 70 or so species of tilapia fish, and they still exhibit remarkable environmental adaptation. So these are kind of differences in temperature, salinity, pH, as well as different levels of oxygen in which they can grow and survive. And so the original genome of Nile tilapia was generated in 2014, which subsequently got improved in 2017 using long-read technology. However, up until that point, that was the only tilapia genome which was sequenced, which of course is not ideal for things like QTL mapping in other species or strains due to fine genetic differences that you can have within the kind of 70 species phylogeny. And so why tilapia, I suppose, as a whole? I think a lot of you here don't really need me to tell you this, but I'll kind of gloss over it briefly. But tilapia aquaculture production has grown exponentially within the last few decades. Nile tilapia of the genus Oreochromis is native to Africa and the Middle East and farmed in well over 120 different countries and territories, and accounts for 9% of the 49 million tons of freshwater finfish produced globally in 2020, making it the 3rd most of any species. What's great about it, it can actually grow and reproduce in a wide range of aquaculture environments. However, the situation being that climate change is leading to extreme weather events, which are then decreasing freshwater resources where Nile tilapia is primarily found. And so within aquaculture specifically, there is then a pressing issue to develop systems based on strains which are resilient to saline water, as an example, as well as temperature changes. And so what is a way forward to kind of deciphering some of this? Well, we can characterize the genetic basis of adaptive traits, which enable selective breeding into, say, farmed strains. And so much of the kind of work that we were doing to follow that up was directed towards this, but more specifically, we were looking at genetic analysis and comparing genetically improved strains against wild tilapia populations to potentially identify genomic signatures of domestication and adaptation which were relevant to aquaculture practices, and more specifically, aquaculture traits. Why is this important? Well, by 2050, the food demand will increase by 36 to 51%. there will be a significant challenge with regards to scarcity and reduced land and water source qualities. And so increasing animal protein demand is important to address, and the growing importance then of aquaculture within this process is important then for addressing that. We've then worked closely then relatedly to that in WorldFish, where they have developed a genetically improved farm tilapia strain, otherwise known as GIFT. Founding population is comprised of wild Nile tilapia from 4 African countries, as well as farm domesticated tilapia from places in Southeast Asia. And this was specifically selected for increased growth as well as reduced population times. And essentially the GIFT can grow 27 to 36% faster than non-GIFT tilapia in these monoculture and polyculture environments, as well as growing with increased body weight. What makes it great is that it's suitable for both small scale and commercial aquaculture. However, what we'd found out from WorldFish and speaking with others, that there is possible introgression of alleles from Mozambicus into the GIFT strain, which nobody had actually examined previously. And so our aim here was to then generate GIFT genomic resources, specifically to identify the genetic basis that could be associated with traits of interest within GIFT. And so a couple of years ago, we published the GIFT genome. This was a chromosome level assembly. I think we got the linkage map from Rosalind. I'm pretty sure we did. And essentially what we're able to find, firstly, this genome's better in terms of completeness compared to the Nile tilapia genome. So covered a lot of the information there. And what we were able to find was that there was 11 megabases of introgression from Oreochromis mozambicus, which basically amounted to around 280 genes, of which those 280 genes were enriched specifically for immunity and growth function. So again, this was really important to create these resources for genomic selection, which can obviously then improve breeding practices and introgression, potentially then at latter stages of desirable traits. And kind of, again, with this kind of same mindset, WorldFish then also developed the genetically improved Abasa Nile tilapia strain. This was initiated in 2001 in Egypt to suit kind of the more Egyptian and African aquaculture conditions. Again, kind of finding these 8th or 9th generation of giant grows 28% faster and heavier than the commercial strain, with almost 38% then of Egyptian farms receiving and then using the 9th generation strain by 2017. And again, having spoken to WorldFish and others, there was a possibility of introgression from a wild tilapia species, Oreochromis aureus, which again, nobody had ever really looked at. So in a kind of similar vein, we generated, again, a giant genome to identify the genetic bases associated with traits of interest in giant, generated a high quality genome assembly, which is comparable to our GIFT one, annotated nearly 40,000 genes, identifying 6 linkage groups, which show regions of introgression from Oreochromis aureus. And we were also able to then resolve the X and Y linked haplotypes, potentially then for sex determination. Again, this is creating useful resources that can improve breeding practices, as well as introgression of desirable traits. And so we just submitted that for review a couple of days ago. But taking all of this together, how can we then further this work to study other aquaculture traits? So generating genomic resources for improved strains is obviously incredibly helpful. Within that, we were able to study traits of growth, immunity, as well as sex determination. But we wanted to look into different methods then of how can we study the genetic basis of other aquaculture relevant traits, and it's kind of more specifically those associated with environmental tolerance. Some studies had kind of delved a little bit into this field. So approaches that they were used involved resequencing populations, mapping and characterizing any levels of genetic variation, then identifying signatures of selection within particular genomic regions which could be associated with adaptive differences. And so these kind of two studies kind of sprung to mind, where they did whole genome resequencing of wild tilapia as well as commercial strains, identifying around 1.4 million SNPs, of which the majority were in non-coding regions. Along that train of thought, only 16 were non-synonymous SNPs, and instead 45% were intronic and 19% were intergenic SNPs, which were obviously in non-coding regions under selection that could be associated with traits of reproduction, growth, and immunity. And so taking together, those two studies found, or what they do suggest, is that selection on functional non-coding regions has obviously played an important role during the domestication and breeding process. And so we wanted to take some of the approaches that they'd used in that study and apply it a little bit further in more of a kind of multi-omics approach. And so again, similarly, asking this question of how can we then robustly identify non-coding selection, but then at the same time, how can we genetically analyze and compare this time instead these traits within wild tilapia populations. And so we embarked on low coverage sequencing then of nearly 600 individuals of 27 tilapia species, and we were able to identify 69 million SNPs along this 27 species tilapia phylogeny. And what we were able to do then was to assess the phylogenomic patterns of recent and ancient hybridization in tilapia. And so we published this just a few months ago. Obviously, a massive problem that's occurring kind of within this space is the instances of introducing species outside of their non-native range without correctly assessing what's in the kind of water bodies already. And essentially what we're able to find is that there's contemporary hybridization between native species and then introduced species. So there's a lot of scenarios of people taking Nile tilapia from one lake and introducing them to another lake, and then mixing of alleles of wild populations and introduced populations, which is then contributing to this contemporary hybridization. However, we were then able to find instances of ancient hybridization contributing to modern tilapia diversity between some of these species. And so kind of going back, almost to remind you, some of my kind of initial studies focused on non-coding selection of regulatory sites and how they could drive diversification across these 5 cichlid species. But instead, this time, what we wanted to do was to apply it to tilapia instead. And kind of up until this point, kind of no study had evaluated the cis regulatory impact of non-coding variation towards adaptive traits in a tilapia phylogeny specifically. And so kind of going back to this point, several studies were able to find evidence of cis regulatory elements that could contribute towards morphological diversity, kind of finding cis regulatory elements at specific genetic loci, whereas epigenetic studies, as I'm sure most of us are kind of well aware, can actually capture chromatin accessibility as well as transcription factor binding, which is active, let's say, in particular cells or tissues at a given point in time. And so some studies had applied this to tilapia, able to look at traits of dimorphism, growth, as well as sex determination. However, they were only able to identify either repressive or active marks at specific loci, and this was not on a kind of whole genome scale. And so I'm sure some of us are quite familiar with the ATAC-seq approach, but if not, it's able to robustly identify genome-wide chromatin accessibility and transcription factor occupancy using very few cells. Obviously now it can be applied to single cells. And so if you're not aware, it allows us to interrogate the open chromatin region. We prepare the material, the cells, tissues in such a way that it exposes the open chromatin, and sequencing adapters bind within these regions, which we then amplify and then sequence. What we're then able to do is that when DNA binding proteins are bound in a particular state at a particular given point in time, like a transcription factor as an example, when we sequence that and pile up our reads, essentially where a footprint or where a transcription factor is bound, they expose this footprint where we can actually then scan for particular motifs of that transcription factor. And so we applied this to tilapia, which at that time hadn't been done previously, so obviously involved developing ATAC-seq kind of within tilapia fish tissues and cells. And what we wanted to then do, particularly with our aim of that, was to characterize open chromatin and the gene expression landscape of Nile tilapia, specifically gill tissue, to identify functional non-coding variation that could be associated with environmental tolerance. And so we started with 3 adult males, dissected the gills and prepared ATAC-seq libraries from those, as well as genomic DNA controls just from naked DNA. We sequenced to high depth for the ATAC-seq per library to identify specifically transcription factor footprints, mapped the genomes, then called peaks in a kind of standard approach. And alongside that, from the same tissue, also carried out transcriptome sequencing to look at gene expression that could correlate with levels of chromatin activity. And so we focus on the gills kind of specifically owing to salinity adaptations that are obviously important for tolerating seawater compared to kind of freshwater fish, such as kind of, as an example kind of shown here, there's the efficient diffusion of water and ions through the gill. And kind of since we previously were able to find that genetic variation disrupts regulatory network interactions in kind of 5 cichlid species, but then, you know, kind of using the Nile tilapia as an outgroup, we wanted to test whether then functional non-coding SNPs could be associated with the regulatory activity of genes involved with, as an example, as we show here, focusing on the gills, of environmental tolerance. And so what we do is we work on the hypothesis here that tilapia species with a similar environmental conditions to the Nile tilapia and tolerance, as an example, would share conserved regulatory genotypes to similarly control gene function. So as a kind of example here of this transcription factor binding site, we would have this conserved genotype throughout, having conservation of that transcription factor binding sites found all along the phylogeny, using Nile tilapia then as a reference, whereas distally related species with extreme adaptations would segregate at the corresponding regulatory site. So we can kind of see here, just instead, it will change from, say, a GG genotype to a T genotype in a particular species. So it would have a different genotype at that locus. And so based on this logic, we were basically first able to associate accessible peaks to the expression pattern of unique genes in gills, identifying actually that most, so 1,650, and peaks nearly 10,500 could be categorized into this high accessibility, high expression group. So these are peaks which are highly accessible and are found in the promoters of genes which are highly expressed. What we then did was we were able to integrate the SNPs from our previous study within those sites. So within the transcription factor binding sites, specifically identifying nearly 1,200 highly accessible, highly expressed genes which have functional variation at the transcription factor binding sites. And then this represents 4% of all Nile tilapia genes and a huge percentage then of the 1,650 genes within that group. So we're identifying genetic variation which could be functional based on this variation that we're seeing at a discrete level within transcription factor binding sites. And so one example of regulatory variation is shown in the gene promoter, then as a kind of expanded here within this prolactin receptor 1 gene, which is specifically involved in tilapia salinity tolerance. And essentially what we find is that variation at the STAT1 transcription factor binding site. So STAT1 is a transcriptional activator that can be phosphorylated via osmotic stress, actually differentially segregates from this TT genotype that basically calls the transcription factor binding site to the TG GG genotype that breaks the transcription factor binding site. And that's actually specifically in this highly adapted extremophile species, Alcalapia grahami. So kind of you could see the genotypes labeled as per the color along the phylogeny, and then the deviation that we can see in that genotype in kind of more than 20 individuals of Alcalapia grahami. And so STAT1, what we then postulate could be a key regulator of the prolactin expression in kind of most tilapia species gills in response to osmotic stress. However, the loss of this STAT1 regulation of prolactin could then aid gill adaptations to extreme alkaline and saline waters that are natural to the alkaline tilapia environment. So those species were isolated specifically from Lake Magadi in Kenya, which is a highly, highly extreme environment, and the color of the water is exactly just like that. And so what we're able to show in this paper was that the generation of kind of novel integrative data revealed certain candidate genes, for example, the prolactin receptor gene, as well as claudin H, which is associated with permeability changes, again, in response to salinity acclimation, and that these genetic relationships and loci could be associated with aquaculture relevant traits like, for example, salinity and osmotic stress acclimation. And so just published this last year. And so I'll kind of coming to the end of my talk now, and what we're able to show in that study is how we could take gene expression and kind of chromatin accessibility data associated to regulatory factors linked to gene expression, and in this kind of gene regulatory network prediction approach, associate regulatory variation with environmental tolerance genes, and more specifically, how discrete regulatory mutations could drive, for example, here, salinity acclimation of the tilapia gills. And so much of the papers that I've kind of worked on in recent years, the kind of grounding for those massively just suggest that selection on functional non-coding regions has played a prominent role on adaptive trait evolution during the kind of domestication or breeding process. And so what's next kind of within the space is to continue to develop systems and network biology as well as machine learning approaches that focus a little bit more within these kind of different areas. So species and population level analysis of more whole tissues, as an example. Some more focused single cell work derived from some of the outcomes of what we're finding from whole tissues, particularly then going on to challenge and stress experiments and comparisons there, especially for species which we know can adapt to kind of a little bit more extreme environments, taking this all down to functional validation approaches such as genetic engineering, but also with the advent of massively parallel reporter assays as well. And this is all within mind to interrogate and focus on environmental and disease resistance traits. And so obviously this work would not be possible without a number of collaborators. Kind of most importantly, I did a lot of this work when I was at the Earlham Institute under the guidance of Wilfred Haerty and some absolutely fantastic group members, and a lot of the cichlid work, which I will continue to do. I'd started under the guidance of Federica De Palma. And as Clarence was saying at the start, I now have a new group at the University of Liverpool. I am a lecturer and principal investigator in bioinformatics, and I kind of want to continue a lot of the work that I'll be doing kind of within these areas of developing computational approaches, kind of more integrative computational approaches, focused on looking at traits of natural and artificial selection, integrative kind of multi-omics molecular profiling, modeling and functional validation, and really getting deeper into these genotype, phenotype to ecotype kind of relationships for adaptive traits, but also continue to look at those which are obviously of relevance to aquaculture. Quite interested, I'll be advertising for a PhD soon as well, kind of applying a lot of these approaches, but a little bit more on the kind of computational and machine learning angle, especially with regards to identifying transcription factor target gene relationships. Happy to talk about collaborations as well as research grants. Happy to take questions. Thank you very much.
So if there's any question in the room, can you repeat them so people online can hear?
Sure.
And I'm just putting this switch to them. Any question in the room?
So I'm really interested in what you showed about the introgression in farm tilapia. And I was wondering, for the contemporary hybridization, do you have an estimate on like the timing of when that happened, or is it ongoing or?
Ongoing. Yeah. Timing is quite difficult because it's hard to know when the species were introduced into the water bodies. And then hybridization with the native that was already in there.
Okay, so it wasn't just that world fish had introduced these.
No. No, these are practices which are almost kind of ongoing within certain areas where it's, I think it's driven through kind of good intentions in a way. Nile tilapia, kind of we know is kind of the third most farmed fish, but the one that everybody wants to eat, and the one that can grow particularly well and to a decent size and is quite marketable in those regards. And so taking Nile tilapia from one lake and dumping it in a local water body is you would think kind of okay practice in theory, because it would feed the kind of local community, but it's not. It's without sampling the kind of natural genetic diversity that already exists within that lake. The problem there being that you have species and many individuals of those species that harbor traits which are really important for aquaculture, which then start to become diminished the minute these hybrids are created. And as we know, sometimes you can have sterile offspring and things like that.
Yes. Thank you for the lecture. You mentioned some parameters that you consider as environmental conditions that you are checking or you are measuring, which include the salinity, pH, and temperature. But have you considered if the species you are checking are farmed species? Have you considered those SNPs that could have occurred due to maybe overnutrition of the water, maybe ammonia load increment in the water, which are also environmental factors, maybe nitrites or nitrates, and which mostly affect farmed tilapia in reality? I think they have more effect on farmed tilapia than the salinity, which people talk most. So what are the considerations for those kind of environmental effects, and have you been able to identify maybe there was a mutation towards that adaptation to those kind of maybe high ammonia load or high nitrite load in the cultural environment?
Apologies, I forgot to repeat the first question, but I've repeated for this one. So the question relates to considerations of the different parameters. So, for example, one's related to environmental tolerance. We've not studied them directly, and so the idea would be to study them directly in kind of isolation, and that's what we mean by kind of challenge experiments. So in a controlled environment, specifically study, let's say, just increasing water temperature, or say just reducing water temperature, and having controls where the conditions are exactly the same. A lot of the work that we've been doing relates to using wild species, and so isolating individuals from the wild, taking measurements of what their environmental conditions are, but we're not specifically focusing on those traits as an example. And more specifically, what is gene regulatory networks doing that could drive some of the differences that allow them to adapt to different environments. Like I said, the kind of next steps would be to see, does that then correlate when we do a challenge experiments? Do we start to still pick up the same changes in, say, transcription factor target gene interactions when we finally start to manipulate an individual parameter?
Great, thanks for the talk, and really plausible. So I don't want to sound like I doubt what is going on. But to start, you said you often find that non-coding regions evolve faster than coding ones. And I guess that's what you generally expect to see, right? No matter whether there's some adaptation in these non-coding regions. And it's totally plausible that a lot of adaptation is driven by perhaps rewiring of regulatory systems. And you found evidence that there are variants that are associated with the adaptation. But do you find this in absence of any coding variation that might also account for this adaptation?
So the question relates to, we see genetic variation within non-coding regions, and is this happening kind of in isolation to variation that might exist in coding regions? Yeah, so that's a very, very good question. And what I kind of go back to is, so those were other studies that found that obviously the non-coding portion was evolving at a faster rate than the coding portion. And I think this is why we like to use the cichlid system specifically for this, because there is literally only 0.1 to 0.25% genetic diversity across the genomes of thousands of species. So we could be taking species from, like we were doing in, say, the 5 cichlid species there, individuals as representatives from each of the lakes, and their protein coding sequences are practically the same. And much of the seminal studies, again, that I kind of alluded to within the 70s were those classic comparisons of human versus chimp, and the protein coding sequences are essentially the same. In 20, yeah, not exactly the same, but a lot of them are the same. And so in the 2014 paper, they did find subtle differences within protein coding regions, which could be driving some of the adaptive differences between the 5 cichlid species, but it was quite minimal. And it was ones where these mutations were obviously not having a deleterious effect. But I would say we've not looked at it directly, but it's a very good question, though. But I would hazard a guess and say all those sequences are going to be exactly the same.
Yeah, continuing of this question, I have one more question. So, exactly based on what Hannes asked, so if you found some non-regulatory elements having evolving much faster, since you had the expression data, you would be able to check their effect on the actual expression level, either in terms of different isoform or something else. So, did you find out those in the data set?
No, that's a good question, but no, we didn't. And that's where I suppose follow-up work on looking at allele-specific expression would be perfect.
And another question which I was going to ask. So, first of all, like I really liked the presentation
Thank
and it
you.
was really interesting. So, since you focus mostly on the cis-regulatory conserved elements across the species to see if there are any kind of adaptation or not, and you could see that. Since you're gonna progress more, I was wondering, you're gonna look at non-conserved part across the species to see over the evolution time if they are really are making the phenotypic differences in terms of adaptation or not? Are there plans for that?
Yeah, so the question was been looking at the question relates to, I was looking at the
Non-conserved.
non-conserved, would I look at the non-conserved part? So a lot of what I've been looking at is the signatures of what is not conserved. So that's discrete variation within these sites. Some of the work that I've done relatedly to, which starts to get a little bit more complicated, of looking at enhancers, and so found kind of say megabases away from genes, and we know that they can act upon them and direct their expression patterns. And some work I've done is to almost categorize active enhancer elements as those which are show evolutionary constraint, so are conserved along the phylogeny, but then those that deviate from neutral model and show divergence. And again, we're seeing a lot of subtle divergence within those. So in answer to your question, yes, and it is coming.
Great, thanks.
Any other questions?
Hey, brilliant presentation. Just a quick question about the ATAC-seq data sets. So you look at the promoter. I was just wondering, did you also look at enhancers and their role in
yeah, so the question is asked, so with my ATAC-seq data, I was primarily focusing on gene promoter regions, and the question was asking whether I looked at enhancer regions. In that study, not specifically, no, because we wanted those relationships of whether activity at gene promoter regions could be directing the expression pattern of the gene, which is obviously just found proximal to the peak. So we didn't specifically look at enhanced regions, but I think like I was saying just there, the plan is to look at those and potentially use contact map information to create the links between whether an enhancer is potentially directing the expression pattern of a particular gene based on topically